So I welcome Madam to take over.
Thank you, Dr. Harish. I, Dr. Prabhavati K, Associate Professor of Department of Biochemistry, Sri Devaraj Medical College, Kolar. Welcome all the participants to the webinar. So I deem it a great privilege to welcome you to the virtual national webinar titled Role of Natural Products and Diabetic Cardiomyopathy as a part of Department of Biochemistry webinar series and to be hosting it. I am thankful to the institution for providing me the opportunity to host this webinar. Now I call upon Dr. Sai Deepika, our Research Associate, Department of Biochemistry, to invoke the blessings of God Almighty. Over to you, Sai Deepika.
सिद्धचारण गण सेवित सिद्धि विनायक ते नमो नमो लंबोदर लघुमीकर अम्बासुत अमर विनोद लंबोदर लघुमीकर सकल विद्या आदि पूजित सर्वोत्तम ते नमो नमो लंबोदर लघुमीकर सकल विद्या आदि
Voice is not audible. Voice is not audible.
Am I audible?
Yes,
Am I audible, sir? Am I audible?
Yes, sir, yes.
So a very good afternoon to one and all. I welcome Dr. Brahmanaidu Parim, sir, for today's presentation. So first of all, I should thank you for accepting our request and invitation to be a part of today's resource person. Our institution, Sri Devaraj Medical College, is a constituent college of Sri Devaraj Academy of Higher Education and Research, which is situated in the Karnataka, east part of Karnataka. And our institution has been distinguished itself as a premier medical education institution across the Karnataka, as well as our institution has got a global recognition with respect to the quality education, patient care, research, and various other academic and non-academic activities that we conduct. So our Department of Biochemistry, every month we conduct a webinar with a research thrust area linked to the fluorosis research, where our institution our department has been conferred with the fluorosis research and referral lab. which has been identified as the center of excellence. We are doing a lot of research with related to the fluorosis. So all these days, there was a lot of research that has been done with respect to the dental and skeletal fluorosis. We are concentrating more on the non-skeletal and non-dental fluorosis, in which we have got a lot of research and publications that we have done. And even we are planning to get a patented instrument where the community health service can be extended at the bedside, which is a point of care testing device. So with this brief introduction about our FRRL lab, so our institution has been started in the year 1986 with the hard work and devotion with a motive to deliver and cater the quality medical education to the population of Kola district as well as across the Karnataka, Tamil Nadu and Andhra Pradesh, because our institution is located at the triangle of these three states. The dedication of the teachers and staff and the vision of our management has led to the noble achievement, to this notable achievement of getting recognition across the globe. Our institution was started by under the dynamic leadership of our late Shri R.L. Jalapaji, who was a central minister, and the cooperation and the extension of the facilities has led us to conduct various academic and non-academic activities. I welcome all the dignitaries, my fellow colleagues, for today's function. I should be very thankful to our respected President of our trust and Honorable Chancellor, Shri G.H. Nagaraja sir, respected Vice President, Shri Rajendra sir, respected Secretary of our trust, Shri G.H. Hanumantraju sir, Honorable Vice Chancellor, Dr. Pradeep Kumar sir, who is a guiding force behind conduct of various scientific activities. I should be thankful to our Registrar, D.V.L. Prasad, where whenever we go with any of the requests, he never says no, and he approves all the activities. I also thank our Dean Faculty of Medicine and our beloved Principal, Dr. P.N. Sriramulu sir, for accepting our request to conduct the webinar with a renowned person, Dr. Brahmanaidu Parim sir. It's my proud privilege to welcome Dr. Brahmanaidu Parim sir, who is a DST-Inspire Faculty, ICMR-National Animal Resources Facility for Biomedical Research, Genome Valley, Hyderabad.
Not audible.
Neurobiology, immunology, and laboratory animal models. He has undergone various training courses in the aforementioned domains, which has enabled him to achieve accolades. He has been awarded with DST-Inspire Faculty and is a topper in MSc Biochemistry and a gold medalist from National Institute of Nutrition, ICMR. His research thrust area in PhD was on isolation of bioactive factors from Piper nigrum and evaluation of its anti-obesity efficacy. He has also worked on the analysis of effect of curcumin on glioma with respect of CD95 and TGF-beta expression. He is a principal investigator and co-investigator for various DST, ICME, and other funded research projects and sponsored projects. He has vast experience in teaching, learning, evaluation of MSc Biotechnology, MSc Food Technology students, and is a question paper setter for various universities across Andhra Pradesh and Tamil Nadu. In addition to the hardcore research and various other activities, he is also actively involved in teaching, learning, and evaluation activities, and is a renowned PG teacher, which has been appreciated by his students. He has authored 4 chapters in the research reference books, which are indexed in PubMed. He has authored reviews and published more than 30 scientific papers in indexed journals with a good impact factor. His presentations have been published in conference proceedings of national and international repute. Once again, it's our privilege and reputation to have sir with us today's program with the title Role of Natural Projects on Diabetic Cardiomyopathy. As I mentioned in my beginning, the very purpose of selecting this topic is to find out the possible implications the fluoride with respect to the diabetic micro and macrovascular complications beyond the skeletal impact, which I have mentioned earlier. I, on behalf of the Fluorosis Research and Referral Laboratory, conferred with Center of Excellence by Sri Devaraj Academy of Higher Education and Research and dedicated team of Dr. Harish R. Along with me, Dr. Munilakshmi U, Dr. Sai Deepika, Indumati Madam, Dr. Soumya N, Dr. Dina Mendes, and Dr. Prabhavati K. I welcome you, sir, on behalf of my department and institution, and your inputs with respect to Fluorosis Research and Referral Laboratory. Collaborating with your thrust area shall enable us to take it further in a long way to go. I welcome you, sir, and thank you once again for accepting our invitation and to be a part of as a resource person. Thank you, sir. I welcome you, sir. Over to Dr. Prabhavati.
Thank you, sir. Now I request our guest speaker, Dr. Brahmanaidu Parim, sir, to deliver the talk Role of Natural Products on Diabetic Cardiomyopathy. Over to you, sir.
Please share.
Yes, sir. I'm listening. Sir, the slides are visible?
Yes, now visible.
Okay, sir. Thank you. Over to sir.
Good afternoon, one and all. Very delighted and grateful to you, and thank you very much for your kind invitation. I, Dr. Brahmanaidu, currently working as a DST-Inspire Faculty in ICMR-National. My core area is diabetic cardiomyopathy. Now here the trend is changing. Here, our major area is diabetic cardiomyopathy. Our lab is mainly focusing on the diabetic cardiomyopathy, how it occurs, what are major pathophysiological changes occurs during the diabetic cardiomyopathy. Sir, am I audible?
Yes, sir. Yes, sir. Please go ahead.
Pathophysiological abnormalities during the cardiomyopathy. What are the interventions are possible with our traditional medicines, especially plant-based medicines. These are our core areas. And here, one moment, slide is not rotating. Role of natural. As you know that diabetes is a situation where hyperglycemic, and this is called type 2, where the excessive glucose will from due to the pancreatic, due to autoimmune disorder is called type 1 diabetes. Diabetes usually 2 types and majorly 5 types, but we categorically say there are 2 types, type 1, type 2. In type 1 context, pancreatic beta cells, it will be destructed during birth itself or self-autoimmune disorder. It's called type 1 diabetes. Type 2 diabetes is induced due to the various factors like sedentary lifestyles, obesity, drug, alcohol, sedentary lives, caloric-rich diets. These are the major culprits induce the type 2 diabetes. In this condition, in diabetic cardiomyopathy, without any abnormal, without any structural changes in the, I mean, without any CAD changes during, sorry, diabetic cardiomyopathy is diabetes associated structural changes. It is not related to hypertensions or caused some other diseases. In this context, patients who are suffering with diabetes, they are, if they consume a tobacco, it will cause us very prone to the diabetic cardiomyopathy. Tobacco consumption is the one of the major cause development of the diabetic cardiomyopathy patients in India, especially in India. Consumption of tobacco is one of the largest culprit causes of the diabetic cardiomyopathy. First is Bangladesh, which is more amount of consuming in tobacco. Secondly, this is not only the this is not only the single problem. Consumption of tobacco and also sedentary lifestyle also one of the cause. The patients who are suffering from diabetes, if they adopted their sedentary lifestyles, it will cause diabetic cardiomyopathy. Every year, due to diabetes, lakhs of people are dying. If untreated, if unchecked, the same number, diabetes 8 millions for 2030. It is estimated that presently more than 8 billion incurred per year for these only diabetic conditions. Nice slide. These are the major pathophysiological changes we are observed in the diabetic cardiomyopathy. One is fibrosis, where the excessive amount of glucose is accumulated in the myocardium, especially this myocardium causes this excessive glucose causes the fibrosis in the left ventricular region, especially. Directly. One moment, sir. 1 moment. Here, fibrosis and increased fat accumulation, oxidative stress, AGE formation, lipotoxicity. Excessive amount of glucose causes the AGE formation, advanced N-glycation products. These advanced N-glycation products directly will bind to the myocardium. It leads to the myocardial dysfunction. Due to the excessive glucose only, these AGEs will form. Lipotoxicity, excessive amount of lipids will accumulate in the heart. It causes the lipotoxicity. These are the microvascular dysfunction, RAS impairment, mitochondrial dysfunction, endoplasmic reticulum stress. These are the major things causes the diabetic cardiomyopathy in patients. Where the insulin resistance is the one of the major cause. Insulin unable to control the total body blood glucose levels. So the excessive amount of glucose, again it will give a lot of pressure to the heart. 1 moment. It will causes the excessive amount of glucose causes this accumulation and formation of the AGEs. Here, insulin resistance one of the major cause, and hyperglycemia, excessive amount of glucose is another causes, and cardiac fibrosis. Generally, it means extracellular matrix in the cardiac muscles. It will accumulate, and cardiac functions are compromising. So initially it will compromise left ventricular region. Slowly it will cause the right ventricular region also, and ejection will be decreased. It leads to the cardiac arrest. Second thing is increased FFA utilization, oxidative stress. Due to the stress also, this excessive amount of glucose causes the stress. Because of this stress, so antioxidant enzymes will be decreased, free radical formation will be increased. These free radicals will accumulate in the myocardial tissue. It causes the compromising the heart function and microvascular dysfunctions. These are the possible. Next slide is that.
Sir, 1 second. I think the slide sharing has stopped. 1 minute, sir.
Slide number 6.
Sir. Yes, sir. Just a minute, sir. I'm just loading it.
Yes. Here, diabetes mellitus. Can you hear me, sir? Can you hear me? Hello. Hello, sir. Sir, my voice is audible. Sir, my voice is audible. Sir, can I proceed? My voice is audible to you, sir. Sir, is it audible? Can you hear me?
Yes, sir, you're audible.
Yes, sir. Can I proceed? Yes. These are the
Yes, sir, please.
pathophysiology of the diabetic. These are the pathophysiological changes of the diabetic cardiomyopathy. Diabetes causes the hyperinsulinemia, hyperglycemia, and hyperlipidemia condition, as everybody is aware and well known. This hyperglycemia and hyperinsulinemia is causes to the cardiac insulin resistance, glycotoxicity, and lipotoxicity. And hyperlipidemia and hyperglycemia situation causes the cardiac mitochondrial dysfunction. If mitochondrial dysfunction occurs, the energy imbalances will be occurs. Our RAS production will be increases. Due to these abnormalities, oxidative stress and impairment of the calcium homeostasis will occur. Once impairment in the oxidative stress and calcium metabolism, then there it will be started the cardiac fibrosis. Once fibrosis is started, cardiac function will be compromised. So there is a impairment of the contractility. First thing is diastolic dysfunction will happen, followed by systolic dysfunction will occur, followed by diabetic cardiomyopathy will be happen. These are next, this side, right side, when diabetes is occurs, this hyperglycemia context is going to happen. This is the situation. There the signaling mechanisms are compromising, mainly glucotoxicity, fibrosis, mitochondrial uncoupling, lipotoxicity. These are the signaling pathways mainly compromising. It causes the diabetic cardiomyopathy. Next slide is that. This is actually where we got funding project. This is the thing we are doing, both polyherbal formulations we are preparing. Then these polyherbal formulation we are testing in both in vitro models and in vivo models. To mimic the human condition, before human trials, we have to go for the animal models and in vitro models. For that, we got from funding DST, 35 lakhs. These are the objectives we are preparing. Initially, we are preparing the phytochemical polyherbal formulation and phytochemical analysis, toxicological evaluation, and dose optimization, both in vitro and in vivo studies. There, rest of the things, these are our main objectives. We have selected these plants, especially Piper nigrum, where we earlier working in this area. Piper nigrum is having anti-diabetic activity, anti-obesity activity, and lipogenic activity. Terminalia paniculata, Gymnema sylvestris, these plants having all the activities like anti-hyperglycemic activity, lipase inhibition activity, amylase inhibition activity. So with that preliminary screening, phytochemical analysis, compound characterization by LCMS and GCMS, we have selected these plants. Based on the toxicological evaluation, dose optimization studies, we concluded for these doses 200 mg per kg body weight. Actually, toxicological evaluation we performed up to 2000 mg per kg body weight. In that, for therapeutic dose, as per OECD guidelines, we have taken 200 mg, 1/10 dose. In Terminalia paniculata condition, 1000 mg we performed all the plants same principle we have followed. 1/10 dose only we have taken. These are the different formulations we have prepared. These are the different formulations we have prepared. These 4 different formulations we were prepared, and we analyzed the toxicological studies. And here next slide is our experimental design. We performed our experimental design. One is normal control. We don't give anything to these animals. This is directly saline will be given. Second one is STZ-NAD. STZ means streptozotocin. If you want to create any animal model, either induced or knockout or genetically mutant model, we have to select. Here, our model is STZ induced model. STZ induced streptozotocin is a chemical which is used for beta cell destruction. Once beta cell is destructed, it leads to the hyperglycemic condition because insulin production will be decreased. So there is no control on the glucose. So excessive glucose will cause us. NAD, nicotinamide. Nicotinamide is another compound which is used for induction of especially cardiomyopathy to mimic the human condition. The patients who are the diabetic who are consuming tobacco, we are not going to give animals directly tobacco. To mimic that condition, we are giving nicotinamide. Here streptozotocin we are giving 35 mg per kg body weight. All these animals are Wistar strain rats. We have internal animal facility. We have these models, and also we have mutant models for this cardiomyopathy. We performed on this on that animals also. For initial induction, we will give from disease control STZ-NAD to up to treatment groups 35 mg streptozotocin, nicotinamide 110 mg. We will give. After 7 days, we will check the vitals, like we will check the glucose. Once glucose level is more than 250, we will confirm this as a diabetic model. Usually it is 120, but frank diabetic, we will confirm as a 250 for animal context. So this is induced model. Here is metformin, the standard drug which is available in the market. We will use for standard drug control. This is nicotinamide alone. Is there any abnormalities like that? We are putting, this is another control. Here PHF1, PHF2, PHF3, PHF4. PHF means polyherbal formulation 1, 2, 3, 4. Earlier slide we are showing these are the formulations we have used to analyze whether these formulations are useful to treat cardiomyopathy, DCM, diabetic cardiomyopathy or not. Here we are categorized the experiment in different parts. One is physiology part. With non-invasive procedure, we will get all these things: total body weight, lean mass, total fat, fat-free mass, fat percentage, total body water, bone mineral concentration, density, ECG, BP. These are the physiological, biochemical parameters, antioxidants, cardiac markers, inflammatory markers. After that, these things are the in vivo. And for in vitro, we collected H9C2 cardiomyocyte cells. These are the rat heart cardiomyocytes. We will be collecting from NCCS Pune. This will be available. With this, we will again dose optimization. Yes, this microRNA, these things, NRF2 knockouts. Our major target is fibrosis, inflammation, oxidative stress, NRF2 pathways. What are the major pathways are involving in diabetic cardiomyopathy? How these formulations are reviving or mitigating the disease? This is our target. So this is our experimental design. We targeted in this way. This is cardiomyocytes, H9C2 cells in vitro. These animal models are in vivo. This is our whole experimental design. Now, once we done this experiment, once we induce, these are the parameters: glucose levels, insulin, HbA1c, BMC, BMD, all these parameters, whatever I mentioned in the earlier slide. We have checked everything. Here, just see group 1 and group 2. Group 1 is normal control in glucose. Here, group 1 is normal range. Here, group 2, STZ-NAD, streptozotocin and nicotinamide, almost 400. So it means it is frankly diabetic. So this is disease control. When we are given metformin, third group, clear-cut evidence is there metformin is reducing the glucose level. automatically nicotinamide cannot influence anything in the after induction of the diabetes. So this is the fourth group is the nicotinamide group. PHF1, PHF2, PHF3. These are showing almost near to normal drug, better than normal commercially available metformin. Fourth one is the fourth formulation. More or less, it is with the standard drug. Same like way, insulin also. HB1AC, BMC, BMD. Here, next I'll go for, I'll come back one slide. Here, BMC, BMD, we are checking, I told you, physiological parameters. Here, just body weight of the animal, 285 grams in normal control. At the same time, disease control, 199. Here, group 4, where the standard drug 218, PHF1, PHF2, PHF3, PHF4, drastically influencing the body weight, lean mass, fat-free mass. Just you check all these numbers. Significantly showing that all extracts are working. So natural products will help to mitigate or revive the diabetic cardiomyopathy. Next, we will go for back. Here, you check the BMC and BMD values, bone mineral concentration, bone mineral density, both the things. Here also, we will clearly observe there is no changes in the normal control. When compared to normal control, disease control, bone mineral concentration is decreasing, bone mineral density also decreasing. It means that calcium metabolism is compromised and bone resorption was happening in the diabetic patients. In standard drug, it is showing better effects. Our extracts, all extracts are showing strong strong bone mineral protection activity. So these extracts will be helpful to treat osteoporosis, osteoarthritic conditions also. It is especially useful for diabetes-induced osteoclastogenesis, osteoarthritis related things. This is the another thing. And also cardiac markers, troponin, myosin, LDH, HSC, these are the cardiac markers. Clear-cut evidence, it is showing our extracts are protecting the animals. This is second thing. After that, antioxidants also. Antioxidants also, our extracts working very better when compared to standard drug. Here, the inflammatory markers, next slide. This is antioxidant engines, already I told you. Inflammatory markers, TNF alpha, IL-1B, IL-6, IL-12, IFN gamma, TGF beta. All the inflammatory cytokines also mitigated after giving the plant extracts. Here we went for metabolomics. Here our metabolomics study. When compared to normal control versus STZ, these are the metabolites: phenylalanine, tyrosine, tryptophan. 1 minute. This is the metabolic studies where we performed a metabolomics at Puttaparthi, Dr. Venkatesh. Here our metabolomic result, when we taken the serum samples of control rats and STZ treated rats. Here, when you check just check phenylalanine, tyrosine, tryptophan, all the metabolites are involving in the hyperglycemia situation. Just these are the glycine, serine, threonine, another metabolites also involving. This is when compared to normal control and STZ. Here, STZ versus streptozotocin, disease model versus commercially available drugs. In this condition also, there are significant pathways. Significant pathways are involving when compared to the standard drug with disease model. Here it is. Next slide. Polyherbal formulation 1. Next slide. Here, top pathways, these are the pathways are involving. When you check the last slide, same pathways are involved almost all. So it means that our extracts are betterly working, are more or less equal to the commercially available drugs. PHF1, polyherbal formulation 1. Here polyherbal formulation 2. there is no significant pathways, only amino acid tRNA, only tRNA biosynthesis is happening in group 2. Same way, polyherbal formulation 3, here minor, only there is no significant pathways, there is no significant pathways are involved in the polyherbal formulation 3. In polyherbal formulation 4, there are few significant pathways are involving. So it means that polyherbal formulation 1 and polyherbal formulations are having diabetic cardiomyopathy protecting activity. So these are the common pathways are involving both the animals and humans. These are the pathways we have taken, and these are the common pathways are involving both animal models and human models. These are the metabolites, common metabolites are involving in both the models. When compared to all the groups, STZ diseased versus standard drug, diseased versus polyherbal formulation, diseased versus metformin, these are the major pathways are involving in the, these are the major pathways are involving. Next is that these are our current work plan is going on. These are the objectives are completed. These are the results we got. We are now started about to start human trials. These are the things where we are going to perform at Puttaparthi. And this is our earlier work where we did with Piper diagram. We got a wonderful result. Here with MRI we have performed. Here the MRI images, it is clearly evidence once animals are induced. This is completely different from cardiomyopathy, where we isolated piperin compound from the Piper diagram seed extracts. From that, we have checked the biochemical parameters and both in vitro, all the parameters, biochemical and non-invasive parameters like body composition, bone mineral concentration, bone mineral density, and where we went for 7 Tesla MRI, Broca's MRI, to identify, to validate the procedure, to detect the fat mass and especially fat distribution in the animals, which may be translation in future. So we clearly got the result. This paper is published in Nature also. This is the one where the epidermal fat around the kidney, it is showing clear-cut evidence. So it means that Piper diagram is having strong anti-obesity effects also. It regulates the adipogenesis, where our treatment, we given a treatment in group 3, group 4 groups. Our treatment is working better than the standard drug. This is the histopathology of adipose tissue, where we can easily identify hyperplasia and fat cell size and fat accumulation. There is clear-cut evidence. If you zoom the slide, you will easily identify. These are our works. Our lab continuously working on non-communicable diseases and dietary interventions, natural products treatment. Non-communicable diseases, we are working on obesity, diabetes, diabetic cardiomyopathy, osteoporosis, osteoarthritis. And also we are working on validation of the natural products, where the India is very, we have lot of herbs which is not scientifically validated. We have 45,000 plant species, around 15,000 medicinal plants are there in our literatures. 15,000 medicinal plants are there, but there is no scientific evidence. So our lab is continuously working on the scientific validation of the natural compounds, especially edible doses. This is our major what slides are why it is rotating. This is our major core area of the research, and we are also doing creation of the disease models, various disease models for diabetes, obesity, cardio-related things, hypertension models, cancer models. We have all kinds of animal models, and we have expertise in the creation of the animal models, validation of the models, and also we are providing CRO for the drugs validation, toxicological evaluation, dose optimization. This is from our end. Thank you very much. Thank you. Over to you, sir. Any doubts? Sir, can you hear me? Please, sir. Internet strength is very poor. Slides, I'm sorry, and I'm very thankful to Devaraj Medical College and sir who is inviting me here today. Some simulation work is going on, bioinformatics simulation. We are unable to connect properly. That is the major problem. Our supercomputer is started and simulation work is going on. Maybe my voice is interrupted. Kindly accept my. Sir, your voice is not audible, sir. Sir, voice is not audible. I'm unable to hear you, sir. Sir, I'm unable to hear you, sir. I thankful to each and everyone who given this opportunity to me. This is my great privilege to deliver a lecture. My voice is, I hope my voice is audible to you.
Sir, your voice is audible, sir. Your voice is audible.
Yes. For creation of animal models, any microsurgeries, any BMD devices testing, we have protocols we will share. We have all the facility, rodent facility, canine facility, monkey facility, horse facility, goat and sheep. We have around 12 species. We have all the facilities. We will provide any kind of assistance, creation of models, and also drug testing.
Thank you, sir. Thank you so much. We are actually looking forward for a long-term association with ICMR-National Animal Resource Facility for Biomedical Research. So looking towards some collaborative research with you. And it is our privilege to have you as a guest speaker, sir. Thank you. Thank you so much.
Thank you.
Thank you, sir. And participants, if you are having any doubts or any clarification, post. Or if you can, if you want further clarification, you can drop in a mail to us. We'll send a certificate of participation to all the participants. So same time, any queries are there, I will forward it to sir, so we can revert back. And now over to Dr. Prabhavati Madam for the vote of thanks.
I request Dr. Soumya to give vote of thanks.
Good afternoon, one and all. Myself, Dr. Soumya, and Senior Resident, Department of Biochemistry, Sri Devaraj Medical College, take this opportunity to propose vote of thanks on behalf of Department of Biochemistry, Sri Devaraj Medical College. First and foremost, we salute our late founder president, Sri R.L. Jallappa sir, who continues to be a beacon of light. We would like to thank our patrons of Sri Devaraj Education Trust, respected President and Honorable Chancellor, Sri G.H. Nagaraja sir, respected Honorable Vice President Sri Rajendra Jay sir, respected Secretary Sri Hanumantaraju KG sir, Honorable Vice Chancellor Dr. Pradeep Kumar Ji sir, respected Registrar Dr. DVLN Prasad sir, Dean Faculty of Medicine and our beloved Principal Dr. P.N. Sriramulu sir, respected Medical Superintendent Dr. S.R. Sheila ma'am, and Deputy Medical Superintendent Dr. Dinesh sir. I also thank Dr. Shashidhar K.N., Vice Principal and Professor at Department of Biochemistry, Sri Devaraj Medical College, for his continuous support and encouragement. I also extend my thanks to other officers of the Academy. I thank distinguished luminary Dr. Brahmanaidu Parim DST-Inspire Faculty, ICMR-National Animal Resource Facility for Biomedical Research, Genome Valley, Hyderabad, for accepting our invitation as speaker and delivering an insightful presentation. Thank you, sir. I thank all the faculty members and stakeholders for their enthusiastic participation in the event. Last but not least, I thank the non-teaching photographer and technical team members who are part of this program. Thank you, one and all.
Thank you, one and all. I request Dr. Harish.
Thank you, ma'am. So thank you, sir, once again. And I now request the participants to please turn on their video cameras so that we can take a screenshot. Okay. Thank you, everyone. And the feedback forms will be shared to the registered emails. So upon submitting the feedback forms, the certificates are auto-generated. Thank you, one and all. Sir, thank you so much. Thank you, sir.
Thank you, sir.
Thank you, sir.